Hello, good morning, and welcome back to the channel. It's always beautiful to have you here. Today, very quickly, we'll talk about what we call biosafety levels. Now, when laboratories are set up, they are set up for different purposes, and there is a method of standardizing laboratories, which we refer to as the biosafety levels. So, when we talk about biosafety levels, we refer to a process of standardizing laboratories and stratifying and classifying laboratories based on the kind of equipment that the laboratory has, the kind of experiments and procedures that can take place in that laboratory, and the kind of pathogens or microorganisms that the laboratory can handle. This is very important because the work that we do in science can sometimes be dangerous due to the different kinds of pathogens and microorganisms that we sometimes deal with in the life sciences. Now, there are four levels of categorization. There's the biosafety level one, two, three, and four. And depending on the country and the place, there are different agencies of government, World Health Organization, Center for Disease Control. Like I stay in Nigeria, so you have the Nigeria Center for Disease Prevention, and then the National biosafety level management agency that's set up by the government. So you have these different agencies that give out these certifications and determine the level at which a laboratory is currently operating and the kind of procedures that can be run in such a laboratory. So, like I earlier said, you have the biosafety level one, two, three, and four. At level one, that's what you have usually at the universities, secondary schools, well equipped laboratories that can deal with different kinds of microorganisms, but not necessarily pathogens. They can handle environmental samples, soil samples, some clinical samples, but at that level, they can't handle serious pathogens. So you see these in universities, in schools, and in different other places, some sorts of research laboratories also. And then the second level at biosafety level two, you talk about laboratories that you have majorly in the hospitals and diagnostic centers and some research facilities of bigger institutions. So you have these kinds of laboratories where they can handle some types of pathogens. They work on their, they do not do much work on their air quality, but they can handle some kinds of pathogens. The workers, the scientists in these laboratories, they wear personal protective equipment. They do a lot of things to protect themselves, but this is at the biosafety level two. Now, the biosafety level three is a more serious level, and here, really serious pathogens, viruses can be handled at this level, and air quality is important to avoid airborne infections, droplet infections and different kinds of infections that can flow in the air. And so the regulations are stricter and they can handle a lot of really serious pathogens. And then the highest level is biosafety level 4, BSL 4. And here is at the top level, and here they can handle anything. It's usually even a standalone building is built for the laboratory, and not only is the environment controlled within the laboratory, even outside the environment of the laboratory, the compound, everything is seriously monitored, and the recommendations can really be stringent. So it's important to understand these biosafety levels as a student and be able to explain them clearly if they come out either in an exam or in an infection prevention control test. Thank you for listening, and remember to like, share our content as we continue to make science easier and more relatable to you. Do have a wonderful day.